Good morning. Indicate your presence in the chat, please. Okay. Mhm. Today, the topic of our lecture is general physiology of sensory systems, Somato-visceral analyzer. We will study the following questions. The first, structure and functional organization of the sensory systems. The second, general principles of sensory systems activity. The third, classification and properties of receptors, reception mechanism. The fourth, the conductive and central parts of sensory systems. And the fifth, structure and functional organization of Somato-visceral analyzer. I'll begin with general physiology of sensory systems. Our organism is an open thermodynamic system. It constantly interacts with the environment. For an adequate interaction with the external environment, an organism must receive information about the outside world and about conditions of all its physiological systems. This is necessary to adapt the body to changing environmental conditions and maintain homeostasis. Analyzers provide the launch of various reflex and general non-specific activation of the central nervous system. Various reflexes and general non-specific activation of the central nervous system. When the functions of sensory systems are disturbed, the knowledge of the outside world becomes difficult. But the interaction of sensory systems makes it possible to compensate for impaired functions. For example, in a person with visual impairment, hearing acuity increases. Also, the study of the sensory system physiology is relevant because the arrangement of these systems can be associated with a wide range of diseases and can use for diagnostic. Environmental information exists in many forms. For example, light energy, sound, chemicals, pressure, mechanical energy, temperature, and other. Our senses respond to a limited range of environmental stimuli. For example, we cannot hear sound of frequencies above 20,000 Hz. Even dogs can hear them. The maximal activity of sensory systems is observed in a person aged 17 to 20 years, after which it slowly decreases. Academician Pavlov, Russian academician Pavlov, proposed a concept of analyzer in 1909. What is it, analyzer? Analyzer or sensory system is a functional combination of structures that provide reception, transmission, analysis, and synthesis of sensory information in the form of a sensation. Sensation is the process by which we receive information from the environment. What types of analyzers? Many years ago, Aristotle described 5 senses like sight, vision, hearing, smell, taste, and touch. In accordance with this, 5 analyzers are distinguished: visual, auditory, olfactory, gustatory, and tactile. And also 5 corresponding sense organs like eye, ear, nose, tongue, and skin. However, in real life, there are more. Also, we have temperature sensation, pain sensation, equilibrium, visceral sensation. With the help of the visceral analyzer, we perceive changes in the internal environment of the body. Also, Pavlov introduced the concept of an oral analyzer, which provides information about the chemical and physical properties of the food taken. Chemicals, for example, taste, physical, temperature, density, and volume of food. Also, we have general sensation and special sensation. General includes somatic and visceral. A special includes smell, taste, vision, and other types. Do you remember links of the reflex arc. 3 links. There are afferent, central, and efferent links. The analyzer includes an afferent link and a nerve center. All sensory systems are organized by the same general plan. According to Pavlov, an analyzer consists of 3 parts: peripheral, conductive, and central. Peripheral, conductive, and central. Peripheral part includes receptors and afferent neurons. Conductive part includes nerve fibers and central part, different structures of central nervous system like medulla oblongata, midbrain, diencephalon mini thalamus, and brain cortex. The peripheral part perceives the stimulus. The conducting part transmits information to the brain centers. In the central part, the analysis of information and its synthesis in the form of a sensation are carried out. What about general principles of sensory systems activity? There are perception, encoding of information, transmission of information, decoding of information, and last one is formation of sensation. Perception is the process of selecting and identifying information from the environment. In the peripheral part, information is encoded, and in the cerebral cortex, information is decoded and a sensation is formed. Processing of information begins at the receptor level and continues to higher brain centers. Analyzers encode the qualitative characteristics of the stimulus, like light, sound, and other, the strength of the stimulus, the time of its action, and the space or the site of the stimulus and its localization in the environment. How is it possible? It's possible by impulse frequency, localization of excited receptors, and by different intervals between impulses. Analyzers are characterized by a multi-level and multi-channel organization. Multi-level organization is manifested in the hierarchical principle of constructing of sensory systems. Multi-channel organization is manifested in the presence of parallel neural channels, which ensure the reliability of information transmission and the accuracy of stimulus analysis. Peripheral part of analyzers is receptor. Peripheral part is receptor. What is receptor? A receptor is a highly specialized structure. It can be cell, specialized cell, or the nerve endings, which in the process of evolution has adapted to the perception of an adequate stimulus. What about properties of receptors? There are specificity, functional mobility, background activity, sensitization, and adaptation. What means specificity? Specificity is maximum sensitivity to an adequate stimulus. For example, adequate stimulus for visual receptors is light. Photoreceptors are maximally sensitive to photons, to the light energy. Adequate stimulus for hearing analyzer is sound. The second property is functional mobility. What is it? It is a change in the number of functioning receptors depending on environmental conditions and the functional state of the body. Background activity means spontaneous membrane depolarization. Spontaneous depolarization of membrane occurs even in the absence of any stimuli. Sensitization of receptors is increase of sensitivity. For example, the sensitivity of the eye to light after 30 to 40 minutes in the dark can increase 20,000 times. Adaptation is opposite property. It's decrease of sensitivity. With the long-acting stimulus, adaptation of the receptors occurs. On the base of adaptation, we can divide it all receptors into phasic or fast adapting and tonic or slow adapting. Examples of phasic or fast adapting receptors. There are photoreceptors, Meissner's corpuscles, Fattor-Pacini corpuscles. Examples of tonic receptors or slow adapting. There are proprioceptors, nociceptors, and other. We can classify different receptors on the basis of different characteristics. The first classification, depending on the nature of the stimulus. There are mechanoreceptors, chemoreceptors, thermoreceptors, photoreceptors, phonoreceptors, and pain receptors or nociceptors. The second classification, by modality. There are monomodal or polymodal receptors. Monomodal, there are exteroreceptors and chemoreceptors. Polymodal, there are interoreceptors. By location and origin of stimuli, there are interoreceptors, exteroreceptors, proprioceptors. Examples of interoreceptors, the receptors in visceral organs, exteroreceptors of skin, and proprioceptors of muscles, joints. Then distant and contact. Distant, like vision and hearing. And contact, there are skin receptors. Next classification, by distribution. General senses receptors, widely distributed, and special senses, limited to head, like vision, hearing, and other. Next classification, by structure. We have unencapsulated and encapsulated nerve endings, unencapsulated like pain receptors, encapsulated like Ruffini corpuscles, for example. And the second type, very specialized cells like photoreceptors, rods, cones, auditory receptors, hair cells, and other. And one more classification, by excitation mechanism. There are primary and secondary receptors. Primary or primary sensory receptors there are olfactory, tactile, and proprioceptors. Primary receptors are nerve endings. Secondary or secondary sensory receptors, there are visual, auditory, and vestibular. There are very specialized receptor cells. What is the receptive field? The receptive field of a neuron is a collection of receptors, impulses from which are sent to a given neuron. Receptive fields can vary in size. Next question, reception mechanism. What is transduction? Transduction is the process of converting energy of a stimulus into neural activity. Our sensory systems work by converting different types of stimuli in the environment, like visible light, sound waves, chemical molecules, into action potentials. What for? Because our nervous system understands just one language. It's action potential. Information can transmit only by traveling of action potentials along the nerve fibers. How it occurs? Transduction. The stimulus changes the permeability of the membrane mainly for sodium ions. As a result, an ion current arises which causes the formation of a receptor potential. Receptor potential, abbreviature RP. Then in primary sensory receptors, the receptor potential reached a critical level and action potential arises, which is transmitted to the central nervous system. That's why receptor potential is converted to the action potential. In secondary receptors, you remember that the secondary receptors, there are very specialized receptor cells. Receptor potential causes the release of a mediator from the presynaptic terminal of the receptor cell. Look at this slide. A receptor cell and end of afferent neuron. Presynaptic membrane, synaptic cleft, and postsynaptic membrane. Transmitter or mediator causes depolarization of the postsynaptic membrane of the afferent neuron and formation of generator potential or GP. And GP is transformed into action potential. And action potential can propagate. Summary. For primary receptors, receptor potentials convert to the action potential. For secondary receptors, receptor potentials convert to generator potential, and generator potential is transformed to the action potential. Both receptor potential and generator potential are graded responses to a stimulus. Amplitude of the receptor potential and generator potential is proportional to the strength of the stimuli. Receptor potential may be depolarizing or sometimes hyperpolarizing. Hyperpolarizing for visual analyzer, mainly depolarizing. The sensitivity of receptors to the action of the stimulus may change. A measure of sensitivity of the receptor is the absolute threshold for perceiving stimulus is the minimum intensity of the stimulus that produce a sensation. Adaptation of the receptors is manifested in a decrease in absolute sensitivity. For example, we don't notice the continuous pressure of clots on the skin. Differential threshold is the minimum difference between stimuli required to provide different responses to these stimuli. Next question is about sensory pathways and central part of the analyzer. Sensory pathways divided into specific and non-specific. Specific pathways there are pathways from receptors to the cortex, to the sensory and associative zones. Non-specific is through the reticular formation, non-specific nuclei of the thalamus, and diffusely into the cortex. Pathways begin from the sensory neuron. The next neuron is located in the posterior horns of the spinal cord or in the nuclei of the cranial nerves. From them, sensory information enters the thalamus. What for? We have double pathways, specific and non-specific. This ensures the interaction of analyzers and reliability of information transfer. Central part of analyzer. Information processing occurs at different levels of the central part of the analyzers. Bulbar, mesencephalic, thalamic, diencephalic, and cortical. The highest analysis and synthesis of information takes place in the cortex. Sensations are formed and the biological significance of stimuli is assessed. Based on this, purposeful behavior is organized. The cortical part of an analyzer is represented by its nucleus or nuclear zone, and diffuse scattered elements. Nuclear zone or nucleus is aggregation of neurons in a particular part of the cerebral cortex. For example, visual cortex, auditory cortex, primary somatosensory cortex, and other. The highest analysis and synthesis of information takes place in the nuclear zones. Scattered elements or diffuse elements ensure the interaction of various sensory systems and partially compensate for the function in case of damage to the nucleus or nuclear zone. In this scheme, you can see organization of the sensory system, peripheral part receptors, conductive part nerves, and central part neurons, different levels of the brain. Sensory means primary and secondary, and associative or tertiary zones are distinguished in the cortex. Primary zones provide the formation of sensations. Secondary zones receive impulses from primary projection zones and provide recognition and generalized perception of signals. The tertiary zones are responsible for the higher analysis and synthesis of information. Cortical projection of analyzers have a topical principle of organization. What it means? Their area is directly proportional to the density of receptors. The sensitivity of the sensory system characterizes the absolute threshold of sensation, is the minimum strength of the stimulus that can cause sensation. Differential threshold is the minimum difference between stimuli required to provide different responses to these stimuli. The relationship between the strength of irritation and the intensity of sensation was described by scientist Weber. The law by Weber. Delta I over by I is constant, where I is the stimulus, delta I is perceptile increment or differential threshold. The differential threshold of sensation is different for different sense organs. But for the same analyzer, it is a constant value. Later, scientist Fechner established that there is a logarithmic relationship between the strength of the stimulus and the strength of sensation. It's Weber-Fechner law. E is equal A times logarithm I plus B, where E is the intensity of sensation, I is the strength of the stimulus or stimulus intensity, A and B are constants that differ for different signals. The laws of Weber and Fechner are valid mainly for average sensitivity range. For other ranges, Stephen established a power law relationship between the strength of the stimulus and the magnitude of the sensation. O is equal K times S in power n. Here must be power n, where O is sensation, K is a constant, S is the intensity of the stimulus, power n is the function exponent. That's all about general physiology of the sensory systems. And last question will be about somato-visceral sensory system. Somato-visceral sensory system includes different types of sensitivity. There are cutaneous sensitivity, proprioception, intra or visceral reception. Somatosensory sensation are the nervous mechanisms that collect sensory information from the body. Its sensation from skin, muscles, tendons, and joints. Cutaneous senses include mechanoreceptive somatic sensation, thermoreceptive sensation, and pain sensation. The mechanoreceptive somatic sensation include tactile sensation, temperature sensation, and pain sensation. Tactile sensation are stimulated by mechanical displacement of some tissue of the body. Temperature sensation detect heat and cold. Pain sensation is activated by any factor that damages the tissues. Let us talk about tactile sensations. There are touch, pressure, vibration, itch, and tickle. Touch, pressure, and vibration detected by encapsulated nerve endings. Itch and tickle detected by free nerve endings. Look at this slide. Here we can see location of different skin receptors. Pacinian corpuscles, Meissner's corpuscles, Ruffini endings, free nerve endings, Merkel's disc. There are skin receptors. Free nerve endings, tactile, Krauss corpuscles, Meissner's, and there are skin receptors and proprioceptors. Tactile receptors. Meissner's corpuscles. Located epidermis, there are touch receptors. Merkel's disc, located in base of epidermis, they are responsible for reception of pressure. Vater-Pasinian corpuscles in deep dermis, they provide reception of vibration. The highest density of tactile receptors at the tips is at the tips of the fingers, nose, tongue, and in the area of a red border of the lips, and the lowest is on the skin of the trunk, forearms, and legs. What is differential threshold of tactile sensitivity or special threshold of tactile sensitivity? Differential threshold of tactile sensitivity is the minimum distance between two adjacent points, the simultaneous stimulation of which causes two separate tactile sensations. For this, there must be one unexcited receptor between two excited receptors. The lower the absolute threshold of tactile sensitivity, the lower the special or differential threshold. Special or differential threshold. Here you see a comparison between differential thresholds on different parts of the body. For big toe, 10 millimeters. For back, 42 millimeters. For forehead less, 18 millimeters. And for first finger, it's very, very small, just 2 millimeters. Conducting part of tactile sensitivity includes specific lemniscal and non-specific extralemniscal pathways. What about lemniscal system? Lemniscal system. Specific lemniscal pathway includes spinobulbar tract in dorsal columns of the spinal cord, and then impulses sent to the medulla oblongata, gall and Buddha nuclei of the medulla oblongata, then to the ventrobasal nuclei of the thalamus or ventrobasal complex of the thalamus. There are specific nuclei of the thalamus. and then to the somatosensory area of the cortex. S1 and S2. S1 is located in the postcentral gyrus, S2 in the cerebral sulcus. The cortical part is characterized by topical organization. Here you can see pathways, fasciculus gracilis and fasciculus cuneatus, and nucleus gracilis and nucleus cuneatus, corresponding goal and Buddha. And cortical part is characterized by topical organization. The projection of the skin surface is carried out according to the point-to-point principle. It means topical organization. The area of the cortical representation of various parts of the body is determined by its functional significance and the density of receptors. Here we can see the sensory homunculus. Look, the hand, especially the thumb, face, especially lips, tongue, have the largest area of representation in the cortex, because they have a high density of receptors and large significance. The motor homunculus looks similar. The second pathway is non-specific or extralemniscal pathway. Impulses sent along the ventrolateral columns, partially to second and the third plates of the spinal cord, to the reticular formation of the brainstem, then to the non-specific nuclei of the thalamus, and finally impulses sent diffusely to the cortex. Information in the spinal cord from most mechanoreceptors comes through A fibers and from T receptors through C fibers. Somatosensory signals from the head travel to the brain via the trigeminal and facial cranial nerves. Next type of the skin sensation is temperature sensation. Temperature skin sensitivity provides information about the temperature of the external environment, which is important for the process of thermoregulation. about the types of thermoreceptors. There are organs of Ruffini and bulb of Krauss. Organs or corpuscles of Ruffini. There are warm receptors. They are located deeper in the dermis. Bulb of Krauss are cold receptors. They are located close to epidermis. There are more cold receptors than heat receptors. Cold receptors are located more superficially than heat receptors. Most thermoreceptors are found in the skin of the face and neck. The conductive part is lateral spinothalamic pathway. It provides impulses from thermoreceptors. The central part includes ventrobasal nuclei of the thalamus, hypothalamus, and somatosensory cortex. If you remember, in hypothalamus, we have centers of thermoregulation, the center of heat production and center of heat loss. Next type of sensation, proprioceptors. Proprioceptors are mechanoreceptors that provide information about the position, deformation, and displacement of various parts of the body. This ensures the functioning and coordination of the musculoskeletal system at rest and when performing movements. Proprioceptors include intravascular muscular fibers, tendon receptors of Golgi, joint receptors, Intravascular muscular fibers or muscle spindles perceive the degree of muscle stretch and are innervated by fibers of gamma motor neurons. The Golgi tendon organs provide information about the degree of muscle tension at which it is stretched, as well as the strength that the muscle develops. Joint receptors respond to the position of joint, the rate of change in the joint angle. They inform the central nervous system about the position of body parts in the space and relative to each other, position of body parts in the space and relative to each other, and about the speed of movement. The conductive part of proprioceptive sensitivity is represented in the following pathways: fasciculus gracilus, fasciculus cuneatus, posterior spina cerebellar or flexic, and anterior spina cerebellar or govers. Look here. Pathways from muscle spines, from the joints, receptors. Impulses sent along the pathways to the brain, And the central part includes the cerebellum, the red nucleus, the lateral nucleus of the thalamus, and the somatosensory cortex. Next type of sensation is visceral sensation, visceral or interoceptive sensation or analyzer or sensory system. The visceral sensory system provides information about the state of the internal environment of the body. This is necessary for the regulation of visceral functions. Also, it is necessary for their adaptation to changing conditions and also to maintain homeostasis. Interoceptors are localized in organs, tissues, vessels, and the central nervous system. These include mechanoreceptors like baro and volumereceptors, chemoreceptors, osmoreceptors, thermoreceptors, and visceral pain receptors. Pathways of the interoceptive analyzer include afferent fibers of the autonomic and somatic nervous systems. Information from interoceptors enters the thalamus, hypothalamus, cerebellum, limbic system, caudate nucleus, reticular formation, somatosensory, and associated areas of the cortex. And last type of sensation of somato-visceral sensory system is pain sensation. The International Association for the Study of Pain define pain like an unpleasant sensory and emotional experience associated with actual or potential tissue damage or described in terms of such damage. Pain is an unpleasant sensory and emotional feeling with available or potential tissue damage. It is physiological sensation and emotional reaction to it. Pain is a primary protective mechanism. Russian academician Anokin said, pain is an integrative function of an organism, mobilizing all the functional systems to defend the body, involving memory, emotions, autonomic, somatic, and behavior reactions to avoid it. Nociceptive or pain analyzer includes peripheral, conductive, and central parts. The pain analyzer gives little information about the outside world. Pain occurs when the body is exposed to damaging factors. The ability to feel pain helps humans and animals to adapt and survive in the environment. The feeling of pain is important for making a correct diagnosis. But the correlation between the degree of pain and the severity of the pathological process is not always noted. Receptors for pain are free nerve endings. They are found in almost every tissue of the body. Nociceptors are absent in the brain tissue, but there are many of them in the meninges. Types of nociceptors. There are mechanonociceptors and chemonociceptors. Mechano and chemonociceptors. Mechanonociceptors are excited under the action of factors that cause deformation and damage of the membrane during compression or stretching of tissues. Chemonociceptors are excited by changes in pH, increases of hydrogen ions concentration when we apply acids, for example, and the action of chemicals or algogens. Examples of algogens, there are bradykinin, lack of serotonin, prostaglandins, P substance, potassium ions, histamine, proteolytic enzymes, and others. Nociceptors also respond to heat and cold effects of higher intensity. The threshold of excitation of nociceptors is higher, while it depends on the personal characteristics, previous experience, social and cultural factors, emotional state, attention, and weather conditions. Pathways of pain impulses. there are specific lemniscal and non-specific extra lemniscal. The specific pathway goes along the A delta fibers with velocity, conduction velocity from 6 to 30 meters per second of the lateral spinothalamic tract to the specific nuclei of the thalamus and to the somatosensory cortex. This is the pathway of sharp, fast pain. The connection between somatosensory and motor cortex provides the motor component of the pain reaction. For example, a protective motor reflex of withdrawing the hand when pricked with a needle. The non-specific pathway goes along the C fibers with conduction velocity from 0.5 to 2 meters per second through the reticular formation to the non-specific nuclei of the thalamus and from there diffusely into the cortex. This is the pathway of slow, continuous, diffuse pain. Here you can see pathways of fast pain and slow pain. Fast pain pathways transmit impulses to the thalamus, and slow pain fibers transmit impulses to the reticular formation, The highest center of the pain is located in the thalamus. The awareness of pain and the degree of suffering depend on the cortex. Also, pain impulses sent to the hypothalamus, limbic system, and other brain structures. This provides the vegetative and emotional components of pain. For example, changes in heart rate, respiratory rate, depth of respiration, blood pressure, and other. Theories of pain sensation. The main three theories: the theory of specificity, the theory of intensity, and gate control theory of pain. Gate control theory of pain. The theory of specificity by Max von Frey postulate that special nociceptors, pathways, and centers exist. The theory of intensity by Goldscheider postulate there are no specific pain receptors, pathways, and centers, and pain arises in overstimulation of any receptors. Gate control theory by Melzack and al. The function of the gate is performed by the gelatinous substance of the spinal cord, which modulates the transmission of sensory information from the primary afferent neurons and activation of neurons of gates prevents the conduction of pain impulses along the spinal thalamic tract. This gating mechanism is controlled by the activity in the small and large fibers. Activation of small diameter fibers opens the gate. Activation of large diameter fibers closes the gate. If the flow of impulses exceeds a critical level, the person doesn't perceive the stimulus as specific, like heat, touch, and other, but person feels pain. All these theories complement each other. How we can classify pain? Pain is classified based on duration, based on location, based on intensity, and based on etiology. The first classification based on duration. Pain can be fast and slow. Fast and slow. Fast pain is sharp, acute, bright, well localized. Felt within 0.1 second if a pain stimulus is applied. It is not felt in many deeper tissues of the body. It can occur in skin and deeper tissues. Slow pain. Slow pain is dull, intense, diffuse feeling, less localized and chronic. It starts after 1 second of application and increases slowly over many seconds or minutes. On the basis of location, somatic and visceral pain. Somatic pain may be superficial and deep. Superficial, for example, on the skin. And somatic deep pain, for example, in muscles and bones. Visceral pain comes from internal organs and blood vessels. Based on intensity. Here we can see pain measurement scale. The first is no pain, and then pain increases, moderate pain, severe pain, and worst pain. Also, pain can be physical and psychogenic. Physical pain is caused by tissue damage, tissue ischemia, and muscle spasm. Psychogenic pain is caused by mental, emotional, or behavioral factors. For example, emotional conflict or physiological or psychological problems and others. According to pain sensations, pain can be described as burning, shooting, breaking, stabbing, cutting, pulling, pulsating, pressing, and other different words. Referred pain. Referred pain is a pain of visceral organs that is referred to a somatic structures. Look at this slide. It's clinical map of referred pain. Referred pain maps are used for diagnosis. What about the theory of referred pain? Modern theory is convergent projection theory. Convergence of somatic and visceral pain fibers on the same neurons in the dorsal horn that project to the thalamus and then to somatosensory cortex. Mechanism of referred pain. Convergence of somatic and visceral fibers to the same neurons of the thalamus, so the dorsal horn, and then project to the thalamus. And brain cortex mistake where is the source of pain. Phantom limb pain. It is a pain felt by an amputee that seems to be located in the missing limb. In our body, we have the antinociceptive system. What is it? It is a set of structures at different levels of the central nervous system that have neurochemical mechanisms that inhibit the activity of the pain system. These structures include the gelatinous substance of the spinal cord, the reticular formation, means the system of descending inhibitory control, also the periaqueductal gray matter, the ref nuclei, the locus coeruleus, the caudate nucleus, the hypothalamus, a limbic system, and also orbital and frontal areas of the cerebral cortex. They synthesize endogenous, endogenous opioids like neuropeptides, endorphins, and enkephalins. Also, non-opioid peptides like neurotensin, oxytocin, vasopressin, bombesin, cholecystokinin, and non-peptide substances like serotonin, catecholamines. The interaction between the nociceptive and antinociceptive systems of the body determines the pain threshold and pain sensitivity. Here you can see picture of antinociceptive system of the brain. Changes of pain sensation. There are hyperalgesia and allodynia. Hyperalgesia, it is an exaggerated response to a noxious stimulus, and nociceptors becomes more sensitive. Allodynia, it is a sensation of pain in response to a normal, painless stimulus. For example, painful sensation from a warm shower when the skin is damaged by sunburn. In clinical practice, we use methods of pain treatment. They include physical, pharmacological, neurosurgical, and psychological. Physical, for example, immobilization, applying of cold and warm. Pharmacological includes many different drugs. Neurosurgical means cut of pain pathways. For example, pathways of definite fibers of trigeminal nerves, because inflammation of fibers of trigeminal nerves causes very severe pain. And psychological, it means psychotherapy, hypnosis, and other methods. That's all about topic of our lecture. Thank you for your attention. Do you have a question? You can write your questions in the chat. Lecture is over. Goodbye.